unmute on your laptop because you're plugged in, I think.
Or I don't know if you should know. Oh, yeah.
Okay. Yeah, so I just have video on here.
Now we can hear you.
Now can you hear?
Yeah, we can hear now. It's
Oh,
good.
Perfect.
Thank you. Thank you.
Your computer is doing the sound. Okay. I'll hide the chat and so with illustrate briefly with some recent work by Ingolfsson on RAS aggregation and response and conformation to membrane composition, simulation-driven work, but where different membrane components change the conformational equilibria of RAS, which is then, of course, a important signaling molecule in both cell state and carcinogenesis. So, why is this hard from a measurement point of view? And it's hard because often one's not looking at a simple, homogeneous, well-mixed system. A membrane by itself introduces an inhomogeneity where there's a solution phase and a membrane phase, but different areas of the membrane may have different environments, in part due to random fluctuations, and in part driven by these protein interactions. And so that is often a key effect in sort of mediating protein membrane interactions. I show here some work on colorotoxin binding and curvature induction in membranes. And so the colorotoxin clustering induces local curvature in the membrane. This is a sort of classic system for this sort of effect. But it's very difficult to measure local effects, local curvature, local composition, because these may be small fractions of the overall population. And so this is the kind of thing that the work in this thesis has made efforts at addressing through a large array of experimental approaches to try to get at this. So in addition to advancing sort of the technical capability for direct measurements, understanding the underlying physical principles is really important. And one of those sets of things is trying to understand what's going on with protein membrane binding for a given protein and membrane partners. And so there are different factors controlling the free energy of association. There's the structural ensemble of the protein bound to the membrane. And then what one would also really like to get at is what the mechanism looks like from a physical viewpoint. And one of the complicating factors is that a large number of what I would say is most interesting proteins exist as not a single structure, but as a conformational ensemble. And that conformational ensemble may change as a function of interaction with the membrane. And that is both something that can be an important response and an important thing to measure from a physical point of view. One of the factors that's thought to control association of many proteins with the membranes and perhaps also play a role in the protein effects on membrane properties is amphipathicity. So this is most classically described for helical proteins, where if one looks at there's very commonly a helical wheel plot, but essentially if one looks at down the barrel of the helix, that one side of the helix may be much more polar and the other side may be much more non-polar. And that property is then thought to permit a certain orientation in the membrane where the nonpolar side is in contact with the nonpolar hydrocarbons of the membrane, and the polar side is in contact with both the polar layer of the membrane and the solution above, which is also polar. And so this property is ideal for interfacial partitioning of a protein and may also drive certain alterations of the membrane, such as stabilizing membrane curvature. And this is something that has been described for alpha-synuclein. Another thing that I think will be discussed is the role of electrostatics. That many proteins bind strongly to charged lipids, and this is exploited synthetically if you want to get a protein to bind really well to a membrane. If you stick 4 lysines on it and use a certain mole fraction of a negatively charged lipid, it will usually associate pretty well. And that's something that if you're trying to stabilize a membrane association, is something that people often take advantage of. But this is also an underlying property in many natural systems, and something that I think we'll probably discuss for alpha-synuclein. And there's an example of that is that the TIM receptors, which bind phosphatidylserine, and they're critical for the binding of a number of viruses, such as Ebola and likely many flaviviruses. And so if one uses phosphatidylserine liposomes, they can competitively inhibit Ebola entry. And so this is, again, one of these protein membrane electrostatic interactions. As I referred to, proteins can often induce membrane curvature. And this is seen with a number of different protein structures, both amphipectic helices, so-called sort of scaffolding proteins, such as bar domains, as well as some other types of proteins that will just insert smaller portions into the membrane. And so this, a number of these structures have higher affinity for highly curved membranes than relatively flat membranes. And so this is an important property of these proteins that is key for a number of cellular functions, but raises a really important question that has been addressed extensively in the literature, is essentially which partner is driving this? Are these proteins that are binding to a flat membrane and inducing curvature, or are these proteins that selectively bind to curved membranes? And I think it's been recently argued that the answer to that is yes, that these are really compatible phenomena and can be described by a single unified physical model, depending on the sort of relative association free energies to these different states, and also to some of the properties of membranes that are highly curved that are also examined in this thesis, such as exposed hydrocarbon defects that might be when a membrane curves, that suddenly the polar layer is a little bit less uniform, and there are more opportunities for proteins that might associate with a nonpolar core to encounter that core than in a perfectly flat membrane. And so that's a key question in this thesis. Another one is binding of not just one copy, but multiple copies of the protein. And again, that's important for curvature induction or curvature stabilization, and can occur via a number of mechanisms, at least in theory, that it could change a change in cooperativity, a change in the maximum number of bound proteins, or a change in the affinity of the proteins for membranes of different shape. And so those are different potential mechanisms. And again, there's some work in this thesis that addresses one of these scenarios. So that brings us now to our star player, which is alpha-synuclein. And again, I'm not the expert in the room on alpha-synuclein, but I will say broadly, there's a lot of debate about its precise role in normal physiology, but it's understood to bind to membranes, and it's understood to play a role in synaptic plasticity, in essentially how the neurotransmitter release response varies over time and can be adjusted. And so that's a key aspect of normal neurobiology, is that one wants to not just form and destroy synapses, but to be able to regulate their activity. And alpha-synuclein is thought to play a role in that. Again, the mechanism is under, as I understand it, long-term ongoing debate. But some of the things that have been put forward are that maybe it plays a role in inhibiting synaptic vesicle recycling. So when synaptic vesicles release neurotransmitters, they then reform and are reloaded with neurotransmitters. And then there's been some debate about whether it has a role in actual neurotransmitter release. I think some people believe it may play a role in promoting the membrane fusion process for release. There's some evidence in both directions on that. And one potential explanation might be also that it affects not the formation of these pores, but the process of pore dilation, which might also speculatively, this is my own speculation, affect the quantity of neurotransmitter release. And then, of course, the reason why a lot of people care about alpha-synuclein is that the process of oligomerization and amyloid formation these physical processes are strongly linked to pathogenesis in Parkinson's disease. And this is a critically important target for those reasons. So with that, I will stop and turn the floor over to our respondent.
Okay, so I don't know if someone can confirm that you hear me on Zoom.
Yes.
We
Okay,
hear you.
great. Yes. Okay, so I just leave everything as it is. Okay, so my first task today is to introduce a little bit the main findings of my thesis. And before I will do this, I will do a short introduction on alpha-synuclein, which is the protein I was working with, and even though Peter introduced, I guess, the most important points. So alpha-synuclein is a 140 amino acid long protein, which is found in vivo at presynaptic termini in neurons. And the characteristic feature of this protein is that it does not adopt any well-defined secondary structure, but it is disordered when it's free in solution, but it is able to bind when it meets a lipid membrane. So the first 100 residues, maximum first 100 residues of alpha-synuclein can fold into an amphipathic alpha helix. And Peter explained what amphipathic means, but I will probably just repeat it because I planned to. That in an amphipathic alpha helix, there is a specific arrangement of polar and non-polar residues. So we have one ideally one phase with non-polar residues and one phase with polar residues. And this is a common feature in membrane binding proteins because in case of alpha-synuclein, we know that the alpha helix inserts a little bit into the membrane, and then the hydrophobic, the non-polar phase interacts with the hydrophobic interior of the bilayer. The hydrophilic phase interacts with the solvent, and it makes sense. And why are we, one more thing. The C-terminal part of alpha-synuclein is very, very, very negatively charged, and it remains disordered even when the protein is bound. And why are we interested in alpha-synuclein interactions with membranes is that there are reasons to believe that the equilibrium between the free and membrane-bound states is core to the healthy function of alpha-synuclein, which Peter introduced briefly. So it's not well understood, and it may be that looking at this equilibrium, at different aspects of this equilibrium, we can shed some light on the healthy function of alpha-synuclein. My thesis is based on 6 papers, but in this presentation, I will focus now on the first 4. So I hope you will excuse me for that. And since in my, most of the experiments that I was doing were involved measuring something as a function of lipid to protein ratio, I will introduce briefly the lipid to protein ratio ranges where we were working. So when we started low lipid to protein ratio, main model system, we used lipid vesicles. So our model membrane system is lipid vesicles, which I tried to show here in blue. So at low lipid to protein ratio, we have excess protein. So all the protein that can binds to the vesicles. The protein that can't stays in solution. So we have 2 populations, bound and free. So if you would go to the right on the lipid to protein ratio axis, adding more vesicles, the proportions change. So you have less free, more bound, and at some point you reach a point where there's just enough membrane to bind all the proteins, but you can continue adding more and more lipids, and then you end up in, obviously, in a regime where you have excess membrane. And one of the questions that we are addressing in one of our projects was, how does alpha-synuclein behave in excess of membranes? And to this, I will get in a couple of slides. But first, paper 1, we wanted to characterize the membrane-bound state of alpha-synuclein. So we were looking at one side of the, on both sides of the equilibrium, but yes, wanted to characterize the membrane-bound state. And it turns out that an alpha-synuclein molecule that is associated with the membrane does not necessarily need to have all of the 100 residues folded into an amphipathic alpha helix. So what this means is that there is more than one possible bound state. So how we get to this? So we were characterizing indirectly the membrane bound state using HSQC NMR experiments. And this is like a widely used experiment in protein science where you, by comparing spectra of protein alone and protein in presence of a binding partner, you can get some structural details on the interaction. And in the case of alpha-synuclein binding to a vesicle, so in this, we have peaks from different residues. When a given fragments of the protein bind to the vesicle, the peaks coming from these residues disappear from the spectrum. So we are basically looking at decreasing intensities, disappearing peaks. And we are interested in the trend of intensity attenuation as a function of residue number. So here we have peak volume for all the residues of alpha-synuclein from 0 to 140 for free protein. We add lipids and what happens? So the intensity changes in a non-uniform way with respect to the residue number. You add even more lipids, you have next step, you add even more lipids, you are just left with the C-terminal residues, which makes sense because these residues are not associated directly with the vesicle. So they stay disordered, we can see them. And basically looking at how these intensities are attenuated, we can distinguish actually at least 3 bound states of alpha-synuclein. So one involving just 20 first residues, one involving 60 more or less first residues, and the other one involving all 100 residues. So, and I will just come back to briefly to our lipid to protein ratio axis. So we are here in this regime. So in order to observe this shorter binding mode, we need to be in conditions where there's not enough membrane to accommodate all proteins bound like this. So when there's enough membrane, so that all proteins can bind with 100 residues, they do so. But when we go to lower lipid to protein ratios, protein excess, some proteins may bind with a shorter fragment. And when we have even less membrane, so some proteins can bind only with the shortest 1 to 20, more or less residue segment fragment. And what this basically means is that by changing lipid to protein ratio, we are not only modulating the amount of free and an amount of bound, but we actually modulate the properties of the bound state. That's the conclusion from here. Moving on to paper 2, we were looking at the dynamics of this equilibrium between free and bound protein. So what we wanted to determine is the exchange rate between free and bound protein. And to do so, we used 2 different experiments. And we are also here all of the time in this regime. Yes. So experiment that we used to determine the lower limit of the exchange rate is diffusion NMR. So it's called pulse field stimulated spin echo NMR experiment, where we look at decaying signal of our sample components that diffuse in the sample. So they decay, we increase gradient strength, slightly complicated, but really interesting. But basically the decay, how fast it decays, so the decay rate depends on how fast the sample components diffuse. And what we were looking at here is, so that circles are decay of the protein signal at different lipid to protein ratios. And for example, lipid to protein ratio 50, we have some free protein, which diffuses fast, so the signal decays faster than at lipid to protein ratio 100, where we have less free protein, more membrane-bound protein, which diffuses slower. And at lipid to protein ratio 200 for our system, the decay of protein and lipid signals overlap, which means that all the protein is bound to the membrane, which kind of confirms all of our other experiments. But this is actually not the main conclusion from here. The main conclusion from here is that the signal decay, as far as we can judge by eye, but not only by eye, is mono-exponential. And this means that in this experiment, we are not able to resolve the diffusion of free protein and of the individual components. So the decay that we see, we have a diffusion coefficient associated with this decay, which is an average of the free and bound states. So basically what this means is that the exchange happens too fast to be resolved in this experiment. And knowing the time at which we actually observe the signal in the pulse sequence allows us to conclude that this exchange is faster than that. And we use 50 milliseconds diffusion time. 1 over 50 milliseconds is 20 per second. So we end up with lower limit for the exchange rate, which is 20 per second here. And we used the analysis of the HSQC spectra that I discussed briefly before to estimate roughly the upper limit. And this is an update for those who read my thesis. So actually at the upper limit, according to our current estimate, is 600 per second. And actually to get to this number, we were analyzing the difference between the chemical shifts that we see in our spectra, which come from free protein, to the literature chemical shifts of alpha helical, of the same residues, but in alpha helix. So I'm happy to answer any questions about this later. But okay, we have a range. We know that the protein exchanges for our system at our conditions, something in between 20 and 600 per second. Yes, next, paper 3. We are looking at distribution of alpha-synuclein in excess membranes. So we actually go to high lipid to protein ratio range. This is where we do our experiments. And the question here is, when we have a lot of vesicles, much more than are actually necessary to bind all the protein, we add the protein and what happens? How does the protein distribute? And we can think of some possible scenarios. So the protein can bind independently to the vesicles, or it can actually bind in a cooperative way. And to distinguish between these two scenarios, we did a really nice, interesting experiment, I think. So this is fluorescence cross-correlation spectroscopy, where we can count fluorescent particles. And experimental system looked exactly like the cartoon. So we prepared blue vesicles and we added red protein. So anything that we see in blue channel, we count particles in blue channel, these are total number of vesicles. We count particles in the red channel, these are only those vesicles that have protein. And as I said, here we are here at very high lipid to protein ratio range. So we add vesicles and we have more vesicles. Makes perfect sense. Yes, experiment works. But when we look at actually those vesicles, we keep protein concentration constant and just add vesicles. But the vesicles with protein their number does not increase in proportion to the total vesicles. So basically it reaches a plateau here. And when we are in this regime, we have actually two populations of vesicles. So vesicles with protein and vesicles that have no protein, which allows us to conclude that alpha-synuclein binds cooperatively to membrane. So we of course did more experiments on this. But coming to paper 4, where we looked at deformation that occurs upon alpha-synuclein binding to small vesicles. So, this we observed by looking at cryo-images of vesicles. So we have cryo-image of bare vesicles, and they are spherical as far as, yeah, they are spherical. You add protein, and they are no longer spherical. So we see a multitude of shapes, unduloids, something like prolates. And yes, and of course, the question that comes to our mind is, what are these deformed structures? And yes, and probably it's not easy to judge on what they are from how they look. But the possible option is that there may be fusion or fission happening, but also an option is that they are just deformed. Yes, but I'll get back to this. But we did an experiment, again, FCCS, fluorescence correlation spectroscopy, where we mixed blue and red vesicles and added protein. And we wanted to see the outcome. We have just correlation curves which report on the number of different particles. We have free vesicles and vesicles with protein. And we wanted to see if we have increase in number of vesicles or decrease in number of vesicles, which would occur if we had fusion or fission going on. But nothing happens. So you add vesicles. So we have some number of red vesicles, some number of blue vesicle, and zero blue and red vesicles. We add protein and nothing happens. So which implies that there's no fusion going on, and there's no or not much fission going on either. So coming back to this image, so we have vesicles, we add protein, but we have the same number of vesicles. We have no vesicles that fused. So what are these structures? An important piece of information here is that we observe these deformed vesicles shortly after, like immediately after adding protein, but we also observe them a long term after protein addition. So here we incubated the sample for 1 day and then imaged it with cryo, and they are still deformed, which is a really important piece of information, because if anyone would like to speculate that these vesicles fused, it makes no sense that they fused. Either they fused and remain in this form for 24 hours, or they just fused after 24 hours, which is even less likely. And this leads us to the conclusion that alpha-synuclein binding provides some driving force for increase in membrane curvature. You go from a spherical vesicle to something like this. And this may not need to lead to fission, and probably doesn't. Yes, this is like a lot of things to say about this, but that alpha-synuclein deforms membranes. That's it increases curvature and it does not cause fusion. So just a short summary: we've been working with the same lipid system. All the experiments or all the results that I was presenting right now were done for the same lipid system and same lipid conditions. That's why I was talking about these 4 papers instead of all 6. So we have some picture. So we know that with changing lipid to protein ratio, we can modulate the properties of the bound state. And there are different bound states, and depending on which on the lipid to protein ratio, where we are, we have different bound states. We were characterizing the dynamics of the equilibrium between free and bound protein, and we got some exchange rate between free and bound. We looked at the distribution of the protein in excess of membranes, and we know that does not distribute randomly. So if you had a lot of membranes, you will have protein-free membrane and membrane with protein. And something that adds to all of it, that you have that alpha-synuclein binding induces deformation. And yes, and that's it. Thanks a lot to my supervisors and everyone who helped me a lot in these experiments. And also, yes, it's very nice to see you all here today. Thank you.
Okay, thank you to both the respondent and opponent for these nice presentations. So now let's take a short break and reconvene here at 9:45. See you then.
set up the camera for the discussion.
All right.
Oh, yes, I guess you could also turn the camera towards.
Yeah. And then
So now it's set to that place, but Håkan is in front of the camera.
yes. Okay, but
Camera is set up. It's up to Håkan.
so we need to tell him to. Can you tell him? Okay, so I will sit.
It's okay, that'd be great to do with the packaging.
Has everyone found their seats? So welcome back to the main part of the defense. I won't talk more. I will just hand the word to the two main actors. Take it away.
Wonderful, thank you. Well, again, I want to say publicly, I very much enjoyed the thesis. I think it's a wonderful body of work, and I wanted to start the discussion by talking about lipid composition. And phosphatidylserine is a common factor in most, if not all, of the membranes you used in your work. I wanted to say, how important do you think PS is?
Well, so I may say first that what we are interested in is probably like a total charge composition. And also in that we would like to have we have a mixture of charged and uncharged lipids that are similar in shape. Otherwise, it will be slightly more complicated system. So so of course I'm aware that this lipid takes part in many many events, but I would say that our interest in it is more like general, that we want to have a charged lipid similar in shape to our uncharged lipid, which makes everything easier. If you want to, interpret data, you have a lamellar phase as equilibrium for your lipid mixture, which makes everything much easier. If we, for example, instead use PE or some fraction of PE, then that would be a much worse choice of a model system if your aim is to have something simple. So that would be my answer.
Do you think, or has this been shown, is there perhaps lower magnitude but similar effect in binding to just straight up zwitterionic lipids?
So what we know is that alpha-synuclein does not bind to purely zwitterionic membranes.
Okay.
So you need to have some charge.
Okay.
Well, there are some studies that it binds to zwitterionic membranes in the gel phase.
Okay, but not.
Where it is probably that it binds to defects.
Okay. Yeah.
So you need to have charge to have alpha-synuclein binding.
And I know you said you're mostly using PS as a sort of as a model lipid, but it is of course enriched in many of the membranes that alpha-synuclein binds to in the cell. Divalent cations can cluster PS. Do you think, would you expect, say, a calcium concentration effect on some of the behaviors you observe?
So I would say definitely, but I need to say that we did not control this in our experiments, but I would expect calcium to both affect membrane and the protein. So this we know. But if we obviously we don't control for it, but we obviously have some calcium in our buffers, because if we don't control for it, we have it at some concentration. So it would make sense, for example, to repeat all of the experiments that I did with controlling calcium concentration, but I'm not up for it.
Yeah, and one I think could reasonably say that unless there's a super high affinity calcium binding, the sort of incidental calcium probably doesn't, you probably don't need EDTA or something around.
Yeah, so
Perhaps I'm wrong.
So I think what's there was some experience by Sarah that if you don't control for calcium, you have something between 30 and 100 micromolar in your buffer. So yes, so well, this would require as much work as I spent doing this to do it with controlling calcium. But we know that like the C-terminal part of alpha-synuclein interacts with calcium and everything. So it's like a really it's interesting and probably important.
Okay, so maybe something for another student.
Yeah.
You when you observe the deformation of vesicles, and you I think it's in the paper, you say you're sort of, let's assume that this is positive curvature induction.
Yeah.
Do you think that there might, there's sort of an attractive reason for that because of membrane defects. Do you think that there might also be stabilization of negative curvature? Or, can you talk about why you stated that? And,
So, I'm not sure if I understand the question.
So I guess the question is, when you show your images of vesicles, and then the vesicles incubated for a while with alpha-synuclein, and you see a lot of these sort of dumbbell shapes.
Yes.
And if I recall correctly, there was a statement in the paper that says, let's assume that the curvature modulation by alpha-synuclein is stabilizing positive membrane.
Yeah, okay, I see. No, I guess. Okay, so by looking at our images, we see vesicles that deform like this, which kind of implies that alpha-synuclein induces positive curvature, and so it increases curvature. I mean, it induces an increase in curvature. And this, and we said that in our analysis we assume that this curvature is induced curvature is positive. And actually, we write that we assume that it is positive, just from some experts in the field, which were reviewers of our article. They say that trivial synuclein always induces positive, has positive contribution to the curvature. So this seems to be obvious for everyone working with this. But there are studies, positive mean, negative Gaussian curvature. There are studies in this. That it induces both of those. But I'm not sure if there's something I can say from our data on this.
Okay. Yeah, no, just the in some ways the induction of those shapes could in theory be consistent with either stabilizing positive sort of around the rims or stabilizing this sort of funny, the interior of the dumbbell, which is, I'd have to think, but I think might well be positive mean negative Gaussian.
So there's a paper that the title is alpha-synuclein that induces both positive mean curvature and negative Gaussian curvature. So yes, probably.
And that could also help explain why you're not getting fission products, because those funny shapes would be exactly what you'd be stabilizing.
That makes perfect sense.
No, they're really interesting results. Sorry, I'm skipping around a little bit in your work, but when you're measuring the helicity or helical content via CD as a function of peptide to lipid ratio, the plateau seemed to occur at a lipid to protein ratio of about 160.
Yeah.
Do you have a sense of why 160? What's controlling that?
Yes, so definitely you would change one lipid in this composition and you would get this a different lipid to protein
Mhm,
ratio. You would change lipids to POPC, DOPS, and you would have it completely in a different range. So, and well, this so first of all, how this increases depends on charge content.
mhm,
But for example, there's a big difference in how this looks for DOPC and POPC,
Mhm.
POPS. This is because like the head group size or area per head group is different, and exposure of the hydrocarbon chain is different. So basically everything that affects binding would, I guess, affect at which point we have the saturation,
mhm,
charge, hydrophobic, temperature.
mhm. So it'd be, yeah, so I guess you'd say all the things that affect,
The protein affinity, I would say.
yeah, although why would that affect maybe I'm just being dense. Why would that affect the saturation point?
Well, so what we know
Mhm,
is that when you take a sample at, let's say, 10 degrees,
Mhm.
you have alpha-synuclein in presence of vesicles, you measure the alpha helical content,
mhm,
you leave the sample in the spectrometer, you increase the temperature. For the same sample, you have higher alpha helical content.
mhm,
And then it's not simply the membrane surface area
mhm,
that governs how much protein is bound.
mhm,
But, well, it seems that protein affinity increases with temperature.
mhm.
So I would say that how like this, when we get the saturation, depends on the affinity of the protein to the membrane. And if you have different charge content, it will it will look different.
Mhm. So temperature effects are, of course, very complicated in membranes.
Yeah.
That normally, if you have a binding interaction and you think there's a temperature effect, you think that's affecting your delta H versus delta S
Mhm.
for binding. Do you think that's an issue, or do you think this is really a temperature-dependent membrane property that's controlling this?
Yes, so we've been discussing this, and this seems to be really hard to tell. So what we do, we just speculate that, of course, like there is a hydrophobic content to binding, hydrophobic interaction strength increases with temperature. There are some changes in membrane properties with temperature, maybe small, but they are, well, many things change with temperature. And I think that we just see that it is like this and we just speculate, but I wouldn't dare to say anything that we think we know what's the origin of this.
Did you, you know, it's a different kind of NMR experiment, but did you think about anything or other, could be an X-ray experiment, depending, I think, but, you know, correlation with tail order parameters?
Order parameter.
The lipid tails, essentially, how, you know, how much the tails are pointing like this versus
so I think
doing
this,
funny orientational
I'm not
things.
sure, but there were many experiments like this done.
Because there may be data already existing.
Yeah,
that
probably. But that was nothing that I did.
Could potentially correlate with this.
Yeah, we've been looking at, yes, but yeah, but there are changes with temperature definitely in what lipids do, right? What the conformational, well, freedom of the chains, head group increases slightly, head group area then. So, well, definitely that I would expect it to affect, but people say that it's a really small effect.
Okay, yeah, I guess, you know, one of the questions is how much of it, this idea of the defects in the hydrocarbon exposure, is that the controlling factor?
Well, so
not?
so I can just tell you how I think about it. So I think about so when we have a fluid membrane, I don't think of hydrophobic exposure as of defects. Maybe it's just how you think about it, but it's just it's fluid. The chains move and the head grouping area per head group increases. You have the exposure. I'm thinking more about defects in terms of when you have a gel phase membrane, and then you have some defects. I mean, but it doesn't matter probably. Yeah, I mean, it's just that we have a hydrophobic and electrostatic component to binding. and that is it. So yeah.
What about, you talk about the sort of PO versus DO. What about polyunsaturated fatty acids?
Yes, so well, definitely. So anything that changes packing of the lipid chains in the membrane, I would expect it all to have an effect.
Because I think synaptic vesicles are enriched in PUFAs.
Well, so you know, we could do many modifications of the composition to try to be more biologically relevant, but it wasn't mainly our goal. But yeah, people do. So for example, there is I call it Cambridge mixture of lipids, where you have some percent of DOP, some percent of DOPS, some percent of DOPE or DOPC. And then, but the problem is that these this mixture is not, the equilibrium phase is not lamellar. And for example, if you want to look at membrane deformation, it's much more complicated system, and probably in all other experiments. So yes, different lipids, I would expect obviously different behavior, and people are looking at it.
Just to ask you to speculate a little bit about physiology. So again, on this polyunsaturated, so polyunsaturated fatty acids, there are data showing that dietary intake of PUFAs then results in direct incorporation in membranes. So what you eat ends up in your cells. So given that, and if this might affect alpha-synuclein binding, and alpha-synuclein binding seems to have then some effect on the oligomerization status, would you speculate that there might be some dietary effects on oligomer formation? There are a number of links here that you can choose which one you want to pick apart.
Well, so this would be very much common sense answer. So I would expect there are dietary effects on everything, and that's obvious. Well, this is pretty hard question, I would say, and I can't say that I ever thought about this, but if this is true, that these fatty acids are getting incorporated in membrane, I see no reason why protein binding would not be affected by this, provided that these fatty acids are incorporated into these specific membranes to which alpha-synuclein binds. I was not looking at oligomerization, so I'm not sure if I can relate to this. But in general, I think as you live and make observations, dietary effects are everywhere on everything. So
I would say that people probably, again, speaking somewhat out of my depth, people probably don't need to be concerned that having fish oil will worsen Parkinson's, because there's also a lot of, even if that would increase oligomerization, there's a lot of homeostatic regulation of membrane fluidity, and the cholesterol content changes. And as you've, I think, discussed, that cholesterol then reduces binding. So I think it probably would negate the effect. So I don't think one needs to go out and.
Well, but it's still good to,
Yes.
Think of what you're eating.
Yes. Yeah. I think one of the really cool findings was the changes in the sort of the length and the region of alpha-synuclein that was membrane associated with lipid to protein ratio. Can you talk about a physical model that might explain that?
Well, so we have an we have alpha-synuclein sequence, and for example, we project it on the helical wheel,
Mhm.
and we see that this separation of hydrophobic hydrophilic residues is better at the N terminal, and it gets imperfect towards the C terminal part. So and yes, and it has been shown somehow that this N terminal most part has highest affinity. So it's never happens that there are some people who think so, but it never happens that the protein binds with residues 80 to 100. But the affinity, if it binds, it starts binding at the N terminal part. So there is like a difference in composition, difference in amino acids in different segments of this membrane binding part, and then these parts have different affinity, and it seems that so what I've been talking about in my presentation is that which of this binding mode dominates depends on the available membrane surface, but it's actually slightly more complicated because it also depends on temperature, because we did these experiments at 2 temperatures and we observed exactly the same steps, but a different lipid to protein ratio content. So when we are at, let's say, 20 degrees, the steps that we see are not. So the reason that at the given lipid to protein ratio conditions, we see this binding mode and that, and not the other one, it's not simply because of membrane surface area constraints, but because also the affinity of the protein. So this is, something probably not that easy to think about. But what we know is, what we can conclude from our data is that this 100 residue fragment has highest affinity. So if there's enough membrane, if the conditions are, then all the proteins bind with this 100 residue fragment. So only when, depending on temperature, and but when you're in excess membranes, for example, in our experiments where we look at distribution in excess membranes, then I would expect that all proteins are bound with this 100-fold residue. Of course, they are dynamically exchanging with protein in solution.
Yeah, I guess what I have a little, maybe the numbers, what I have a little trouble wrapping my head around is if you think they're, sort of simplify to say there are, a certain number of membrane binding sites, and the N terminus has the highest affinity.
Yeah.
And then is the model that at a lower lipid to protein ratio, the N terminus of another alpha synuclein is competing with the sort of mid residues of the first one for those available binding sites. Is that the model, or?
Yes. Yes. So one thing is that looking at our NMR data, we can get more or less fractions. And even at low binding, at our low lipid to protein ratio conditions, most of the proteins are bound with all alpha helices. But there's some fraction, for example, for example, there's 20% that are only bound with short fragments.
Okay, so it's a minority. Okay, because what I was thinking is that, the problem with that is unless your solution phase alpha synuclein is at huge excess, that once you're bound, the local concentration of the next few residues is going to be super high, right? Because it's right there.
Yeah.
And so you'd have to have a really high excess of solution and terminus to compete with that.
We have, but looking at percentages, for example, at our lipid to protein ratio, like low, the first step that we observe is that more or less only 20% of the residues are bound with the short fragment. The rest are bound with full fragment. And then when we go to higher, we have just 10% bound with shorter fragment, and then the rest. But of course, if you have a whole membrane, it affects the properties of the bound membrane, even if it's just 10 or 20%.
Yeah, so it's still the majority,
yes,
which would then be consistent with this sort of competition. Okay, thank you. I had this stupid technical question, just my lack of understanding. But for the pulse field gradient, stimulated echo NMR.
Yeah.
The diffusion time was fixed at 50 milliseconds.
Yes.
How much leeway is there in that?
How much what?
Leeway, how much could you change that?
Oh, so this took us a long time. So, yes. Okay. So, so there was a lot of optimization, and so there are a couple of parameters that you can adjust to get good data, meaning no artifacts. And for example, diffusion time or gradient field strength, maximal gradient field strength are one of the parameters that you can optimize. So what we want is that we want to set the maximum gradient field strength and the diffusion time so that in the end of experiment, we get high decay of the signal. And it was a challenge for our samples because so we measure free protein, we measure protein at with a little bit of lipids, with more lipids, so it's completely different. So the diffusion properties are completely different. So we wanted to have conditions at which for all our samples we observe enough decay to get use of our data. But with this, the important thing was that for the conclusions of our conclusions, it was important that we have the same diffusion time for all samples. Of course, you can measure whatever, but we were interested. So when doing this, we were interested if we will see a difference between different lipid to protein ratios. So we don't. We see mono-exponential decay as far as we can judge for all of them. But and the parameter that we were modifying was the maximum gradient strength. But we started. So the idea was to start with shortest possible diffusion time, which I think was 20 milliseconds or even less. But then we were getting artifacts, and it took us some time to optimize this. But it actually was, I don't think it's necessary for our conclusions to go to this super low. But for example, if we were measuring sample at lipid to protein ratio 50 and we had 20 milliseconds diffusion time, we also saw one exponential decay. Yes, so it was just that for you can play around with this parameter, but for our conclusions, it was important that we have the same diffusion time for all samples that we want to compare.
Yeah, no, because I think as you were alluding to, if you could adjust it and measure successfully, that could help you bound the exchange times.
Yes, definitely.
But it
so the point is that it took yes. So but when I did it, we just focused to have good data and good enough data and for all samples so that we can compare it.
Like, talking about exchange times, you've made a lot of progress in trying to figure out what the bounds are on the exchange time. So let's start by sort of thinking through. So what is an exchange time?
So it seems that people define it as time for one on-off event. So whichever is slower, the on or off. So I guess that we say that it's 20 per second.
Okay. Yeah, I guess I would have thought, so if you think about sort of, decay times for like pre-steady state kinetics, and you have an on and an off, the 1 over exchange time would be 1 over K off plus 1 over, if you have quasi first order, 1 over K on times whichever concentration is in excess.
So we were discussing this, but I guess so of course you have K on depending on concentration, K off and stuff like this, but it's like from the experimental point of view, I guess that what we measure is the time for like the time scale on the on-off event.
But you're measuring the time scale to come to steady state, right? Not just
We-
one on, one off, right? But
So we are at equilibrium.
yeah.
Yeah.
Do you, so, and then I guess expanding on that, if there are potentially multiple association modes,
By that you mean many proteins going on and off, or
well, I don't know, this maybe starts to get into, do you
Yeah.
think that the first step of association is always the same? Or when you're getting, binding in some of these different modes,
okay? So
do you think
okay.
that, do you think there's one on rate here or multiple on rates?
Okay, one disclaimer: I don't understand chemical kinetics,
Okay.
unfortunately. But the other thing, oh yes, of course. So, so we can expect that we have on-off exchange, but we
Mhm.
can also have exchange between the different binding modes,
Mm,
which complicates
yeah.
everything. But for example, and as we were discussing, so we measured this, we were estimating this diffusion NMR experiments we did at different lipid to protein ratios, which are in the ranges where we expect to
Mhm.
have different binding modes dominating.
Mhm.
And it makes sense to expect that the kinetics, the dynamics can be different in
Mhm.
different regimes, but this is nothing that we can resolve with our experiments.
Yeah.
But the NMR experiment is done at lipid to protein ratio 200, where we expect to have all proteins bound with the full helices.
Mm,
And for example,
mhm.
a lipid protein ratio 50, where we have some bound with short and some bound with long. So definitely we would expect to have the exchange between the different binding modes.
Mhm.
But I think this is not what we could, I would assume. So in the diffusion experiment, we look at diffusion. So the free protein diffuses like free protein, and bound protein, I would assume that regardless of whether it's bound with short or long fragment, it diffuses with the vesicle.
Yeah.
But it would be quite interesting to know what are the dynamics of the exchange between the different binding modes. And there's one paper where just based on HSQC experiments, they concluded that the exchange between different binding modes is also slow on the NMR chemical shift time scale as the
Mhm.
exchange between free.
Mhm.
But this, I don't remember what was the motivation for this.
Which do you think is slower, the exchange between free and bound, or the exchange between the different binding modes?
Well, probably free and bound. Because if you have the, for example, you have
this is not a question with
yeah.
the
But you have let's assume that you have the protein with bound with the first, I have no idea, right? But if you have the protein bind with first 20, it doesn't need to diffuse to the vesicle, it doesn't need to meet it doesn't need to meet it in the right conformation, it just slides and binds. So it's already there, and for my understanding, I would assume that would make it faster. But
and the idea also from your HSQC is that it's gonna is it? Do you have helix formation breaking in the different binding modes, or?
yes, I guess I would assume that it's bound, it folds. This part is unbound and it folds. It like I think people
It's sort of
think of it
helix
like as a
extension.
zipper.
Mhm.
It just unzips or
Yeah.
zips.
Okay. Can you think of a way, so all of these are exchange times, and the exchange times are obviously hard to get at, as you've.
Yes.
Is there a way that you could, we talked about yesterday some
Yeah.
other potential experiments, but I was also wondering, this is not a question necessarily where there isn't the answer, but is there a way that you could get at, say, K off, even if you couldn't get at exchange times?
Yes, so there are quite interesting papers looking at peripheral peptide bindings to membranes, and this was something with fluorescence. So something was changing with fluorescence upon binding, and then doing experiments in many different concentrations, and from this. Yes, so then people are extracting K off, for example. They do also experiments at different lipid different vesicle sizes, and they judge, okay, if it's diffusion limited, if it's depending on what's the relation of the KO on to the size of the vesicle. So yes, I guess that, but this was actually done for alpha smoothing. So that was something that I would have done if I didn't do many unnecessary things for 4 years before getting to realize this. But this fluorescence, like kinetic, I guess, experiments, stopped flow fluorescence, that's what I mean. I think, I'm not sure if I probably should refer to this in my paper. I don't know if I do, but they got results consistent with ours. I actually not sure if they were taking it down to K on and K off, but probably yes. But just doing at different concentrations and, you know, some fitting of the data. I don't know how reliable that would be, but people do this.
Yeah, or load and wash out, and
Yeah.
depending on your mixing time. Okay, cool. Sorry, let me just check. Oh yeah. I thought the analysis of binding cooperativity was really cool. And I had a couple questions about some things that were striking and a few things that just confused me a little. So when you're looking at this was, I think, paper 3, figure 2C or something, but don't. But when you're looking at the brightness per vesicle, and you saw that increase and a peak and then a decrease to the plateau.
Yes.
What do you think is causing that?
Yes. So yes. So one thing. So for your conclusions of this paper, what is interesting is that from some lipid to protein ratio, there's not much change. But I think this peak in brightness is consistent with our different binding modes, because then you would have more proteins per vesicle, which would give a brighter vesicle, and that it is not high from the beginning, but it increases then probably because at the lowest lipid to protein ratios, there we have free protein, like free protein plus bound protein. And at some point when we are at this high brightness, I would expect that then we have maximum number of protein molecules per lipid.
So help walk me through this. So we have the protein favorable interactions that are the cooperative binding.
So this would be this is our guess, right? So we just speculate on the molecular origin of cooperativity, but I think it's fair to expect that.
I'm just, so let's take that for the moment as a given. So you have the protein favorable interaction. You have the protein lipid favorable interaction of binding for different segments. And so the idea is when there are a fair number of lipid sites, but not vast excess, then the competition between those two interactions ends up with a larger number of proteins bound because they end up bound with a shorter segment.
Exactly. And I think that this was our aim, to actually be able to show this with FCS, looking at brightness. And it's there. We don't bring it up in the other paper, but I guess that this explains why we have higher brightness, and then it decreases. And I think that what we discussed recently is that the fact that we have these different binding modes, and that the longest binding mode is the one that has highest affinity, makes sense in the context of cooperativity if attractive protein-protein interactions are important, and probably they
Mhm.
are, then the longer the helix, the more if there are
Mm,
helix attractions, then the longer the helix,
mhm,
the more of it you get. So we kind of think that this is consistent, that this binding
Mhm.
mode has highest affinity, and when proteins can,
Mhm.
they bind like this. Yeah.
yeah, I guess that it depends on whether it's helix or also with the
So
Uh,
probably there are different other
yeah.
contributions from like changes in memory properties or from dissolvation or whatever, but we could expect probably
Yeah.
that attractive protein-protein interactions
Mhm.
play an important role.
Yeah. No, and I guess that's one question, is this is the cooperativity direct or indirect? And you have this for the amyloid formation, if I understand correctly, you postulate sort of a head to tail binding mode.
Can you explain this more?
I thought, maybe I misread, that for the amyloid formation, you were suggesting that the C terminus of one may
Yes.
interact with the N
Yes. So okay.
terminus
So
of
now I
another.
see. So this is. So first of all, we don't know what happens,
Mhm.
but there is some experimental data and there's some simulations that show that if you have surface grafted with C-terminal part,
Mhm.
which can either be a surface of a fibril or surface of a vesicle covered with protein, then even though the C-terminal parts are negatively charged
Mhm.
and the protein is not negatively charged,
Mhm.
the fact that you have inhomogeneous distribution of charge in the protein sequence causes that you have some attractive interactions between in this ordered protein between the charge, the N-terminal of protein and the C-terminal, which is grafted bound on the surface. So this is, I would say that that's all we know. There is so the protein aligns in some way when there's not too high ionic strength. And I guess that but what happens actually then, I guess we don't know this.
Do you think that might also play a role in the binding cooperativity, or do you think that's likely a different mechanism?
Well, so according to everything that I done and read, it seems to me that when all the protein is bound, I wouldn't. I mean, of course, there must be interactions between protein-free in solution and protein bound, but I wouldn't say that this is necessary for cooperativity. I mean, you have proteins on the membrane and they are fine. I guess that's important for what happens in the long run. I mean, how,
I guess
don't know.
it's just, if that's a stabilizing interaction, it could be some fraction of that, sort of, free energy, delta G, that is estimated for cooperativity.
Yeah.
Or it could be totally unrelated. It's if it's a stabilizing interaction that could be operative in this case, then it could be a component of that. Of course, if that's either there's a CN interaction or there's a N membrane interaction, but not both, if those are competing, then maybe that's a different mechanism and not playing a role.
Can't tell.
It's a complicated system. It's I did dumb question about the cooperativity analysis, trying to make my way through the equations.
Okay.
And if I understand correctly, the equations treat the first, second, third binding site.
Yes.
Per vesicle.
Binding event, I guess.
Binding event. Okay. Yeah. And I guess this was sort of my question is I wasn't sure if, you have one bound and, if it wasn't clear to me whether if you end up, potentially saturating a number of sites, that when one's bound, you would think that the second binding, unless it has to be in one particular binding mode, that there's a combinatorial effect where it could bind at a number of sites.
I'm not sure if I understand the question.
Okay, so if you think about, what we could look for chalk.
So definitely there are statistical effects. I
but,
mean, if you have 10 proteins binding, 10, for example, vesicle with 10 binding sites, you have statistical effects that the binding constant for the first, second, fourth is not the same, even though the binding is independent.
Okay.
And this is easy to calculate because then, yes. So there, yes. So if you would like to get some binding constants, I guess, for first, second, fourth protein bound, so just not the same number. Because of the statistical effects that, okay, if you bind the first protein, you have 10 ways to bind it. If you find the second, you have 9 ways to bind it, and 2 ways to unbind it. So then you get, from the statistics, you get binding constants for independent binding, and yes, and to calculate or to, yes. So the binding. So anyway, so if you have some, if you have one binding constant for example, second protein bound and one for the first, and you calculate the delta G has a component from statistics and component from the interaction.
Okay. Yeah. Okay. Yeah. That's what I wasn't seeing, and I was worried about.
Yeah. So it's there, probably not so well explained, but
it's taken into account.
So the average affinity is the same for both independent and cooperative binding. I guess that's the idea.
That the delta G is sort of the protein effect from first, second, third, fourth, and then you also have the combinatorial effect also.
Yes. So it's just if we have let's say 2 binding constants in the case of cooperative binding, and we want to calculate delta G, we need to take into account the statistical effects. I mean.
No, it just looked like the model had a constant, 10, and I was like, wait a second, there should be this combinatorial.
Yes, definitely we have it, and I think that you can in figure legends and in the methods we write what are the constants for independent binding, and then the constants for cooperative binding we calculate it so that there's the same free energy coupling per each binding step.
Yeah,
because it doesn't need to be so, but I think we did that. We have 10 kilojoules per mole between each,
but then that's before this combinatorial effect is
I
taken
think this
into
is
account.
I would expect, I think I would need to check the equation, but I think it's minus the combinatorial effect, because I think so, or not, but there's
okay. Yeah,
but I think that this free energy coupling has the component from pure statistics and from interaction
and from
that we
okay, cool. If one considers alpha-synuclein to bind, changing gears a little bit, to synaptic vesicles, those are kind of small, and they have a lot of protein in them already.
Yeah.
So, would you expect the sort of big, long binding mode to predominate, or in those cases, do you think maybe it's more?
Well, so I guess that would depend on the surface of this vesicle with respect to the place that a given helical fragment takes. So I kind of, it's hard for me to imagine like right now some ratios of this. Well,
Yeah, no, you don't have to come
Um,
up with.
but anyway,
Mhm.
so we would expect that if you, it's probably not the answer for this, but we would expect if you have a vesicle packed with proteins that at least this our cooperative, not our, the cooperativity is manifested by binding in patch.
Mhm.
You don't have excess membrane, but like we would expect that it's manifested by binding in patches, so that protein
Mhm.
binds in patches instead of randomly. And what is the
Mhm.
binding mode? I mean, probably we can expect that the cooperativity is strongest for the full helix,
Mm.
but we can't say that it's not there for the shorter ones, I guess. Mm.
Yeah, no, and again, not knowing the physiology enough that people sort of talk about this as binding to synaptic vesicles,
Yeah.
but whether it's to the vesicles or to, synaptic membranes.
Yeah, so I guess
Um,
this can be, yeah, this is an interesting thing to
Good.
look into.
It would seem if you have this 40 nanometer object with a bunch of stuff in it already,
Yeah,
it would be hard to imagine there being a lot of binding sites, but maybe.
yes. But I think that for the understanding of, I guess, everything, we don't need to have plenty of membrane.
Yeah. Well, and I think, as in a lot of these complicated membrane physiology cases, one can study the physical interactions in model systems that are more straightforward,
Mhm.
that might not exactly reproduce the physiology,
Definitely, that's what
but
we're
the
doing.
interactions
Yeah,
are still there in the
Yes.
physiological system.
Yes, definitely. That's, I guess, what we're doing.
Yes. Yes. No. So putting all this together, alpha-synuclein exchanges rapidly between the solution and membrane-bound forms.
Yes.
The bound, broadly speaking, conformational ensemble depends on the protein to lipid ratio.
Yes.
And binding to membranes also depends on the sort of cooperativity of binding to membranes also depends on the protein to lipid ratio. It might have a higher affinity for curved membranes. And you have evidence of membrane deformation. Is that a
Yes.
reasonable summary? Leaving
Yes.
aside the aggregation in amyloids for the moment.
Yes, I think it makes sense. So thinking from the last point, like membranes that deform, proteins that deform membranes bind cooperatively. This makes sense. And this was nothing that this is not the knowledge we started with, because we just saw this, we just saw that, but it seems to make sense. Well, the fast exchange between free and bound is completely consistent. So alpha-synuclein is peripheral membrane binding protein. And in this case, so I will say nothing specific about any specific protein, but in case of this peripheral membrane binding proteins, the whole idea is that it binds transiently and it exchanges fast. And if you take some proteins, that peripheral membrane binding proteins, you do something, you mutate the protein so that it binds stronger or less strong, you have some bad effects for the function. So also the observational cooperativity, we in our experiments, we saw non-random distribution of protein in excess of membranes, and it was there. We didn't have to wait 2 hours for this to happen. It was there. It was established at that time of the experiment. And in our cooperativity paper, we hypothesized, okay, that the exchange rate must be fast, even though at that time we didn't know what it is. In order to have this, in order to this non-random distribution to be established in the that time of the experiment, we expected fast exchange. So I would say that cooperative fast exchange deformation it all makes sense. I guess the presence of this binding modes kind of complicates it for thinking, but it doesn't contradict anything. I mean, probably it just involves more thinking. But you know, so it's not that I'm not critical to what I'm doing, but it kind of makes sense in the end, even though it, you know, was not like this all the time.
Yeah, no. And I think it's a nice, I'm asking, can we sort of assemble a picture?
Yes, I think we can. So yes. So I was thinking to put the cartoon at the end of my presentation, my thesis. I didn't, but it kind of, you know, we have it for one system, for one conditions, which is also an advantage, because, you know, people work at different conditions, and then maybe it's slightly harder to relate these experiments without repeating everything. So we have it. We looked at different aspects for the same system, which is simple. And I think that, yes, this is an advantage of this, I would say.
It's a fascinating and complex molecule. Wonderful. Well, thank you. I have no further questions.
Thank you very much.
Okay, thank you for that nice discussion. So now it's time for the questions from the examination board, and I suggest we start on Zoom. Do we have a link to Gothenburg?
Yes.
Thank you.
Okay. Well, first of all, I also want to say that I really enjoyed reading your thesis. I think this is a very beautiful piece of work. And I think you answered a lot of questions that from the opponent already that I had been collecting. So I'm just going to have to look for a few questions left here that we still can ask you. And I think something that was kind of touched upon already by the opponent, of course, I'm interested in the neuroscience perspective of this. And this protein that's so prevalent in the synapse, and we know it seems that it's involved in synaptic activity somehow. And some research is suggesting that synuclein is affecting plasticity and fusion pore activity. Then we already mentioned this about calcium. So you said that you have some calcium present in your system right now?
Yes.
Yes, and what concentration do you estimate for your calcium?
Yes, so this is a subtle point. So we don't control for calcium, meaning that we neither add calcium nor add EDTA. But knowing from some estimates that were done around in the lab, if you don't control calcium, you use glassware, prepare buffer in a glass flask, you have calcium. So this and there is just an estimate that it's something like 30-100 micromolar. I can say if I have 30 or 100, but there must be some calcium concentration, which we did not control for, and it would be a good idea to control for. But it would be probably clever to do it from the start.
Yeah, exactly.
To have the experiments at the same conditions. So we know that calcium affects aggregation, I guess. We know that obviously it may bind to the C-terminal part, which would, screen some charges on the C-terminal when the C-terminal part, when the protein is bound to the membrane, C-terminal part sticks out, some calcium binds, it changes the properties. And thinking about this, it may change many things that we looked at. But I can't also say that, it will be all, if we, add some more calcium, or that it would be different world. Probably not. I mean, it obviously, what we have must hold, but definitely it would make sense to control for calcium somehow.
So do you know what the calcium concentration is in the cytosol?
I don't know. I could check.
So it's in a nanomolar range.
Okay.
So and when calcium acts through synaptic activity, do you know what concentration range?
No.
Okay, because I was going to say, because then you end up in the millimolar range. So basically, you are already kind of way below what usually triggers action. So I think this is interesting too. Okay, but then when things happen in the cell, because the cell is, as we know, full of membranes and there's membrane dynamics going on. When neurons ask these synaptic vesicles to fusion form the fusion pore, there's also gradients of membrane tension involved. So what do you expect for alpha-synuclein to respond to membrane tension in lipid membranes?
Okay, so this is not an easy question, and I think we've been discussing and thinking of what actually membrane tension is and what it does. So so it will not be easy for me to say what I would expect. I actually know what what tension changes with other proteins which deform membranes, that it changes binding, changes how the proteins bind, for example, in the case of dynamine or the other proteins. But I think this is a really hard question to say. And I need to also say that I've been looking into this and trying to understand what membrane tension is, and I didn't find it easy.
Okay, because I was wondering, like, this cooperativity binding, is membrane tension involved in that process? But maybe that's an aspect then you haven't really thought about.
So definitely we were thinking about this, but I think in our systems there, I think from what we did, there's I at least can't say anything, but that would be definitely an interesting thing to look into.
Yeah. And I guess the opponent also talked about this. It's an interesting thing, like where, because inside the cell and in these biomembranes, the protein content is already very high. And I was also thinking, where on earth on the synaptic, very fully packed membrane can this little protein bind? So, and then I was thinking even more about it when some research talk about affecting the fusion pore. So if we think about the alpha-synuclein binding to a 40 nanometer synaptic vesicle, the fusion pore is about 2 to 4 nanometers. So there we have some kind of very high curvature, and there's both negative and positive curvature involved. So do you think that a tiny protein like this would actually have some type? Can you speculate like how this protein maybe have some fördelar?
Okay, so do you mean how can such a tiny protein have an effect?
Yeah, exactly.
So
yes.
I guess the answer is that it does not act alone. That the proteins act together. And that would be my answer, and that is I in many membrane deforming proteins the case that it's not one protein that binds and deforms the membrane, because it's in many cases that would not be that would be hard. It would be hard for the protein to provide so much energy for such a change. So I would expect that anything that alpha-synuclein does to change the membrane curve, the membrane properties, curvature, shape, is done by many proteins binding together to some membrane patch, and not one single protein. Yeah, so that would be my guess.
Yeah, because we know that there's these kind of complex snare protein complex docking the vesicles and binding. So there's this whole protein machinery there already. So I was just wondering, like, if you could see it like synuclein being like the essential key, like fitting in the keyhole in the machinery or something like that.
Well, Well, so yes, so regardless of what this snare machinery does, what it does, yeah, it docks the vesicles, right? So alpha-synuclein has an effect on its own on the membrane properties.
Yeah.
And yes.
Yes. And I know this is not the focus of your thesis, so I'm stretching the thoughts a little bit. But then I have one very basic question too, I think. So I was looking into the experimental section for all the work that you've done. And I see that in all experiments, you have used a 10 millimolar mass buffer at pH 5.5, so a switcher ionic compound. And so I was thinking, how did you motivate using this buffer, and considering, like, does it have any, so two things, does this buffer have any effect on membranes, and what motivated your choice of buffer, the concentration and pH?
Yes, so this is a longer story, but maybe starting with the concentration of the buffer, it's low, because we want, there are electrostatics in our system, and we want to see what that it does. We don't want to screen. We don't add 150 millimolar sodium chloride. So low salt, because we want to see, we have electrostatic interactions and we want to let them be. Mass buffer, so the pH 5.5 is we used it because, so actually originally I guess it was that there were studies done on alpha-synuclein aggregation, which is what I started looking at, even though it's not the biggest part of my thesis. And that these conditions at mildly acidic pH, secondary nucleation is playing an important role as compared to what happens at different pH values, which is actually not never mind. So this pH was chosen for that reason. So we know, so starting to look at aggregation, we know that secondary nucleation, I think oh my God, but in this case, secondary nucleation is on the fibril. Here we have heterogeneous primary nucleation, never mind. But this was, yes, I mean, there's a difference in what happens in aggregation when you have a neutral pH and mildly acidic pH. And I think what is important right now is that if you have vesicles at pH, let's say 6.5 or 7, and vesicles at pH 5.5, vesicles at this neutral pH do not cause aggregation. And when you are at this mildly acidic pH, you have the aggregation. And probably that was the reason why we used this buffer. So the protein is binding to vesicles both at pH 5.5, obviously at pH 7, but the effect on aggregation is completely different at this mildly acidic pH. And this is what I said already. And apart from this, there's like, it kind of makes sense. So here there are many people who work on this, and we're working on similar conditions, which kind of makes sense because we can later compare our data. Yes, but I think probably the answer should be that the effect of vesicles on aggregation is different at different pH.
Okay, I see.
I think.
That's the motivation for it. Okay.
Yeah.
Well, I think I'm happy with those questions. Thank you so much.
Thank you.
Okay, thank you. So, Mikael, would you like to continue?
I so much enjoyed reading this thesis.
I'm happy.
In particular, the summary.
Yeah.
It's very nice. It reads very well.
I'm happy.
It's mechanistic and nice. It's not too much detail.
Yeah, only the essentials.
I love that. Dobra robota.
Dziękuję.
When I read this, I think about the cell studies, but I don't want to talk about physiological relevance or anything, because I hate that what people do, because obviously all these things are physiologically relevant. It's a matter of how it pans out.
Yeah. Mm-hmm.
I don't know where to start. You do your experiment at the relatively low concentration, a fraction of
Concentration of
when you look at cooperativity.
concentration of protein, you mean?
Yes.
Yes. So different experiments are done in different concentrations. So the FCS is at low concentration. Probably it was something like, or maybe 250 nanomolar protein. Yeah.
So it's relatively low compared to the cellular levels, which is say 20 to 50.
Yes. But for example, we do cryo at high concentrations. Cryo is like 15 millimolar vesicles, lipids, and then accordingly less protein.
And you see then, I mean, your pictures here, where they have the different binding modes.
Yeah.
You really coat these vesicles. I'm looking on page 82 here. Fractions, and it's one bit that binds first, and then you go more at the end of the day. In your high, I mean, where you have only the negative tail sticking out.
Yes.
And that would, of course, I don't know. I ask you. I wonder that would really change the surface property.
Very much. Yes. So yeah, this is what we speculate on in this paper, that that of course the existence of the different binding modes leads to modulation of membrane surface properties, membrane covered with protein by lipid to protein ratio. And looking at in this summary, I put this simulation snapshot from page 52, right? So that you have interaction of a monomer in solution with the protein covered surface, whatever this surface is. So having these different binding modes definitely affects this interaction. In which way, it will be hard to say for me right now, but we expect it plays a role, and we know, or we think that it is the membrane bound surface of the vesicle that plays a role in the aggregation and presence of membranes. It's not free membrane. And then having the possibility of having different properties of this membrane surface, it's an important piece of information, I guess, to have in mind.
At the same time, now you're changing this. It was difficult to follow exactly in your different experiments. In your paper, what the state of the vesicles. So as I take it, the system is also very stable. You do aggregation studies over weeks.
Yes, it takes time, and it's also at 37 degrees, the aggregation that we did. So at 20, where we did most of our experiments, is super slow.
Yeah. But still, it indicates that this vesicular system it's a very good colloid, so to say.
Yeah, negatively charged.
Can you induce flocking in this system?
I don't know.
Because I've seen with that, because if you were to cut out your first positive little bit.
Positive part of the protein, then I'm not sure if it will still bind. So I guess how much, depending which fragment you would cut out.
But if say you take the part with the electrical density where most of the positive charges, wouldn't you expect that to bind on its own?
No. I mean, if you cut out the part that binds, then I would not expect the rest to bind.
No, but I mean, if you make a fragment of only the positive.
Yes, so that, okay, so the C terminally you truncate it. Okay, so definitely this would bind, and I think it was shown in some experiments, different people, that this aggregates faster. And the idea is that the C terminal part has some protecting effect on the aggregation. And also, probably I mentioned this somewhere, that the C terminal part has some transient interactions with the membrane bound part. So definitely this part binds on its own, but things are different, right? Because you don't have this part that is negatively charged. It changes probably the interactions between proteins on the membrane. Yeah.
Because we've seen in other systems, not we, but
Yeah.
colleagues doing,
Yeah.
sport with some positive charge, and they interact in a way which is similar to yours.
Yeah.
Forming helix that has some hydrophobic embedding, and then you have some positive chargings coordinating or according to the structures to negative limits around.
Yeah.
And then you see that the system easily flux as well. I mean,
Okay.
salt and so on.
Mm-hmm.
That you have that. So I wonder if the rest of your system is then somehow protecting the system from undergoing
Well,
that kind of reactions. And then you have a what you would call a binding mode where you have the hydrophobic bit. Is that right?
exposed.
Hanging out.
Yeah.
Yeah, that would give some hydrophobicity to it, and then eventually you have only the negative part hanging out that also interacts with calcium, for example.
Yes.
I noticed in your on page 137
Mm.
where you have a nice picture where you show deformation and the deformed vessel.
137. I don't have this page.
Sorry.
Ah,
Oh, 134. Sorry.
okay. There's no page 137. Never mind. Yes. The cryo figure.
Yes.
And what do you say?
The ones that you have made red there,
Yes.
they are the ones with the protein in them.
Yes.
And they are a bit deformed, but they're also co-localized.
Well, so with the so yes, so I would say that I'm not sure about this. So I can't say by my eye if it's random or not. But in sometimes when things appear random, they're actually not. Or when never mind. But in cryo, so this is like a slightly different system. But I spent some time trying to label alpha-synuclein with gold nanoparticles, which was hard. But then you have your sample is super viscous, and then you can pipe it up and down all day. But when you look at the cryo image, you have like almost hexagonal arrangement of vesicles in the sample. So I mean, I don't know here if I would say, but there can be issues with cryo. But I wouldn't say that. Yeah, but it's hard to say for me if this is anything.
With the tail up, the negative tail up.
Yes.
You would expect to drop this. I mean, just thinking about the cellular concentrations of calcium.
I mean, of course, I mean, you screen the electrostatic repulsion.
The ion interacts with
Yes.
the negative part, so
Well,
that's promiscuous.
Mm.
Sort of like states,
Yes.
I don't know.
Yep. Definitely. Why not? But probably that would be good to check.
Then going to people have done in Selenol.
Yes, I know.
Perspective, and then you crank out or you electroporate it.
Yes.
Whatever you do, and I think that's also interesting that it's electroporated. I will come back to that. Then what Phil Selenko, for example, did it first, because you have really nice narrow language. So I mean, the initial result was that there was no change on this protein.
Okay.
The in-cell spectrum were basically like a solution spectrum. And you really had to look hard for any change. So that suggests that the protein, if you put it into the cell at some, I suppose it will back to the range, doesn't bind at all.
Yes, so I guess there are other experiments that people say that it's predominantly disordered free in the synaptic environment.
And then people also, when they internalized the protein, that was, or at least in some of the cases, I don't know all these slides in detail, but that typically operations, so then it would cause a lot of membrane. you would upset the membrane in the process with the pores. And
Yeah.
so on. And then one would imagine that why doesn't the protein bind on the way? You see what I mean? It
But
shows
okay.
high affinity and so on. Why?
But then my question would be, so you introduce some high concentration of protein into the cell. And can you actually tell by looking at your NMR data that can you actually rule out that some of the protein is bound, but because you don't see it, but you can't quantify it? Yeah. So that would be enough for me to understand. I mean, I would say, right? So I don't know how quantitative you can be. Okay, that there's small fraction that is bound, completely consistent with everything, and the rest is not because I don't know, there's no place or whatever.
So it could be in there, you say, but it's hidden because it's a small.
You know,
fraction
so
of the internalized protein.
yes. So in our experiments, it's clear we have some. We don't have a cell. We have just a test tube buffer. We lose some protein. We see it clearly. We lose the peaks, right? We know, okay, it's bound. I just don't know how quantitative it is and how much bound protein you could resolve, how big change in when you're in animal.
It's tricky. Because
Yeah.
it's always, you don't know how much you get in. It's 20. It's not
Yeah.
homogeneous also.
So, of course, this is much more complicated system, but I would consider that, things can be going on anyway, as you would expect the protein to bind, but probably would you expect all the protein to bind? That would be maybe not, right?
It's just sort of, to me,
Yeah.
looking at your results, I would have guessed that you would recruit a substantial amount over time, at least, because this is when it takes time to express the cells. You have the electroporation, so you would present membrane sections, and your binding is relatively strong here. Most cooperative, one would expect that it goes in, and maybe it does.
But can you try to electroporate different amounts of protein and find the lowest amount where you have some data? I mean, you are able to.
This is not, or at least not in our hands, it's not a strict science.
Mm.
You try and you make the
Yeah.
proteins, and then you're happy with
Yeah.
the protein in them. And of course, protein leaks out as well, which is another interesting
Yeah.
factor here.
So this is super interesting. So this is interesting, but if you have a small fraction bound, then
Maybe someone should look at that actually.
Yeah. Mm.
No, I also noticed that your NMR results here, where you see that the first part of protein,
Yeah.
you really have an interaction. That's what people now in the aggregation studies, they see that by introducing chaperones into the cell.
Mhm.
Binds to the very same root.
Okay.
Yeah.
Mhm.
Whatever trigger factors in
Mhm.
70s, they have exactly the same effect. So
Okay.
whenever you bind here to the membrane, it seems always,
Oh, that's interesting.
which is interesting.
Yeah. Mhm.
Can you, but you can do these vesicles in the synapse, and have you tried that to do extract physiological vesicles?
So people, we didn't do this, but people do it.
Yeah.
You have some brain extract, and then you maybe someone did it here. I don't know, but we did not do it. Yeah, but
It's all very. I have a
Yeah.
maybe very stupid question.
Go ahead.
It's about curvatures.
Yeah.
I understand how we argue, and I buy it for.
Mm-hmm.
You choose not to talk about the cooperativity in terms of protein structure, and I would like to. I will not do that.
I don't know what you mean. I mean,
No,
I think you can talk about whatever.
The mechanism of the cooperativity.
Yeah.
You don't go into the structural details, but you.
Yeah, because so I can just say that that this is an ongoing project, and this was not we saw it, we characterized it in some way. We have hypotheses about what can play a role, and now there's a student working on it, trying to look at whether you see cooperativity if you change conditions, high salt, different pH. So there are there's ongoing work trying to figure out what's the molecular origin. But this is, I can say whatever, but I don't know.
No, I have much more basic questions.
Yeah. Yes.
relating to curvature,
Yes.
maybe silly.
Of course.
Of course, you can, if you have what is called defects,
Yes.
you will expect expand the surface.
Yeah.
Like you showed in your initial picture with the mushrooms in the coat.
Mhm.
But then imagine now that you have what is called a bicell. That is very big.
Yeah.
It's a pancake.
Mhm.
It's this big and very thin.
Mhm.
And then you add membrane proteins that do not go into the membrane, but they sit loosely electrostatically associated. So they don't really use any hydrophobicity. They're just bound.
But can it be? Okay.
Hypothetically.
Yes.
And then you have proteins there. Then, I mean, if you have an ideal gas, so I mean, you could have a concentration there that would try to increase the surface area.
Concentration of
I mean,
bound
just having
protein
the
on the
proteins there would
membrane.
put some sort of pressure, if I express it badly. Would that curve a membrane just
So-
by having a protein concentration? Because without interfering structurally with the membrane, but just by that, because they want to then try
I
to
think
improve
so.
it would.
I think so. So, you know, I read some books and then there's, you know, equations for anything that can bind, absorb, and cause curvature in some way. I don't know if you can have it without any hydrophobic interactions, but I would say yes.
So that if that is present to whatever level, that would in your experiment favor a spherical particle.
Can you?
It could favor an elongated one with a bigger surface area as well.
So we see that in different lipid systems, when you don't have vesicles, but you have slightly different things like discs, like disc micelles or something like this, you see the formation of this when alpha-synuclein binds. And there were also papers where from some lipid composition alpha-synuclein were deforming it into something like a bicell, really long things. Yeah. Thank you.
Yeah, thank you very much.
Okay.
Well, like the others, I've enjoyed reading this. I think it's very nice to read,
Thanks.
and in particular in the summary part. I like your use of boxes, the explanatory boxes. Very helpful for someone more of
You know,
side.
it was meant for you. I mean, not only, but
Okay, well,
but
good for me at least.
I mean for everyone, but like for everyone to enjoy without, you know.
Yeah, it broadens the readership, I think.
And
Because
that was the idea.
In particular, since they explain a bit of methodology.
Like
things
what?
about.
So the idea was to explain what you need to know to understand the data and not everything or whatever.
But it's a good model that I think others should adopt.
Yeah, I really like it.
Well, I should then explain that I'm a neuroscientist working with Parkinson's disease from an experimental point of view, and my questions will reflect my interest in synuclein and its role in pathogenesis.
Yeah.
So,
I mean, you're
it's
welcome.
a little a different angle from your own. So there are two things that are very much in focus of that research, and one is of course, the toxicity and the formation of fibrils, aggregates.
Yeah.
And it's all in. And the other is the role, the progression of the disease and the ability of the disease to spread. So my first question is, can you comment a little on how your mechanisms could explain the transfer of pathogenic forms of alpha-synuclein?
So I think I would say that my results, I wouldn't think that my results contribute to anything, to any better understanding of this. unfortunately. Yes. So so I think
could lipids or membrane play a role in the actual transfer process? Because it's been seen either as a transfer at the synapse,
yes.
so through synaptic contact, and the other is the possibility that it is non-synaptic and interacting with cells in the neighborhood, for example, through exosomes.
Yes.
How could that explain?
So yes, so what I could say is that if alpha-synuclein, I think we know is associated with the synaptic vesicles, which release their contents of the synapse. And if, I guess it's not hard to imagine that alpha-synuclein in some form could be passed on together with the things that are released at the synapse. So I guess this, I cannot say that I have anything to cover what I'm saying, but this is kind of something that I would be able to believe in. But whether, for example, it is inside some exosomes and is transported, I would say, why not? Probably there was research into this. So it can be transported via vesicles to which it binds.
Okay. One thing I'm curious about is when is affecting the curvature of the membrane.
Yeah.
Is it on the outside surface or inside?
So so in our experiments, alpha-synuclein is added to the outside of the vesicles. It binds to the outside, and this affects which way the curvature changes. Yes. So yes. So obviously, yes. So if you have from which way it binds, it affects how the curvature, which way curvature is changed. So, but if, for example, we have a synaptic vesicle and alpha-synuclein binds to the outside, we could expect that the effect would be similar to what we see in our experiments, because what we know is that monomeric alpha-synuclein does not cross the membrane. This is what we know. There are data on what oligomers do. And this is actually not something that I was looking in, but there are data that show that it can be different for oligomers. So the behavior of oligomers, whatever they are, well, obviously is different from the monomer. And this is a completely different story. But the monomeric protein, as far as we know, as far as we see, and as far as our data is concerned, does not cross the membrane. Which obviously affects how it affects the membrane, because if you would have the protein bound on both sides of the vesicle, then you would, with the same coverage, you would expect no effect on the membrane curvature or shape. But the fact that it binds from one side, it's what makes it, it's like the asymmetry in the membrane that makes it bend.
Yeah, do I understand correctly that you don't believe alpha-synuclein is involved in fusion, vesicle fusion, but in fission?
So I will clarify this. So I don't believe that alpha-synuclein on its own causes fusion. So I know that there are these snare complexes and everything, and alpha-synuclein takes part of it, and I have no comments for what the general state of being in biology is. So but people claim that alpha-synuclein alone induces fusion, and we think based on all that we did and all our data and how we understand it, that it's not the case. And this, I would say, is clear. But this has nothing to do to what alpha-synuclein does with all of the other machinery. Because there were ideas that alpha-synuclein on its own bridges vesicles, makes them fuse. So this is what I don't agree with. Yes.
But could one draw a conclusion that alpha-synuclein is more involved in the uptake process? We think of that communication between cells could be vesicle.
Yeah.
Would it then be more on the uptake side than on the delivery side if it's
Definitely.
so it could play a role in the ability of the cell to pick up the material when it's
Yeah, definitely. So one thing that I was reading up on is that when you're at the synapse, at the presynaptic terminus, you have pools of vesicles that are somehow waiting for their turn, and they are somehow, they need to be they are kept by some proteins waiting for their turn to, you know, to do something. I mean, to take some cargo and release. And what is interesting, it turns out that there are some proteins, I think this protein is called synapsin, that it clusters the vesicles. And this is considered to be liquid-liquid phase separation phenomenon, where you just have a formation of some dense phase, which in this case is composed of vesicles with protein. And alpha-synuclein, so this is pretty new view on the subject or on what happens. And it seems that synapsin does the job, but alpha-synuclein is also there. And this is mostly, I would consider this relying on some non-specific interactions that these proteins have. I mean, they just cluster the vesicles, and upon some stimulation, whatever it is, I don't know, change in something, concentration of something, they release these vesicles. So, and like the fact that alpha-synuclein is disordered in solution kind of makes sense in this, because this is what disordered proteins do. They are able to undergo this phase separation. So this is like an idea. So these are probably quite recent findings. Yeah, but this is for me quite makes sense. Of course, I don't have like a broad understanding of what happens, but it makes sense for me.
That's interesting.
Yeah.
My next question is related to the formation of protofibrils, which is very much again in focus in the mechanistic reasoning and how pathology.
Yeah.
So you use a model system with some vesicles you produce yourself.
Yes.
So question is, what do they represent inside the cell? Could you say that you're modeling? Obviously, alpha-synuclein doesn't bind to all membranes in the cell, then it would be totally over-decorated.
Yeah.
So what do they represent? What would be the counterpart inside?
So I think that the vesicles that we use, we want them to be not too big and charged, which, for example, synaptic vesicles are. And that's all we have. We have some charge so that the protein binds, and they are not huge. They are not big membranes, flat for the protein, or maybe they are flat. So I think that the idea is to model synaptic vesicles. But we don't, but of course we could do a better job in modeling the composition of synaptic vesicles. But this comes with a cost that if you go to something that is more complicated, then it's just harder to interpret your data. Because like by adding some more lipids that are, that you would be happy that, okay, this is really synaptic composition, then you can induce things that that are really, how to say, that will make everything slightly harder to interpret some, which is what people do. And so by comparing data of other groups, it makes sense to think how does this lipid composition actually affect what happens. And our system, so of course we have simple systems, and for our systems, you know, it still is complicated, right? We have the protein binding, it can bind in different ways. When it binds, it deforms the membrane. And many experimental techniques, so one of the things we wanted to do some specific NMR experiment, phosphorus NMR. And then the fact that the membrane is deformed and that the shape is different completely compromises. It was super hard. We just left it because it was hard to get the information to deconvolve everything. So yeah, so I would say that our idea, and many people that work with alpha-synuclein vesicles, the idea is to mimic synaptic vesicles in more or less size and composition. But what we want to do is we just adjust the model system based on what question we have. So we saw some behavior with one lipid composition, we change something to see whether the composition or whatever that is changed affects what we see. And I think that that would be our approach most of the time, that we have a question and this defines our approach, which includes also the choice of model system.
Yeah, that's an interesting suggestion, because it has some bearing on where the formation of protofibrils take place.
Yeah.
And when looking microscopically at that, it seems that it takes place in all parts of the neuron. So it's in the cell body, in the dendrites, in the axons and axon terminals, which would suggest sort of nucleation places that are spread.
Yeah.
And synaptic vesicles are actually a good candidate for that, because they are formed in the cell body and then distributed throughout.
Okay.
So that's an interesting suggestion. Now, when it comes to formation of protofibrils and the relationship between lipids and synuclein concentration, you describe in one paper how the relative concentrations play a role.
Yes.
Is this strictly membranes, or is there a role of the sort of free lipid molecules?
Yes. So for our lipids, they have very, very, very low solubility. They are just very hydrophobic, not very soluble in water like oil. And for our purposes, we can assume that we have, I think for our purposes, we can assume that we have no free lipid concentration. So all we look at when we have this lipid to protein ratio, it is, of course, we have a membrane which has an outer and inner part, and the inner part is alpha-synuclein is not interacting with, but we think of the membrane of the vesicle, not about free lipids in the case of this lipid system. But of course, you can have lipids that are much more soluble.
What
Fatty
would happen
acids.
if you have soluble lipids? Would that
So I think there has been work done on interaction of alpha-synuclein with fatty acids, which I guess are lipids that are much more soluble. Are they? Yes. So I'm not sure. I would need to read up on this, but that would be a completely different story. Yes.
One reason for asking is that one of the clear factors that determine the propensity of alpha-synuclein to form protofibrils and aggregates is the concentration of alpha-synuclein. So the endogenous cellular level in the cytoplasm. And when I read the paper on the relative proportion, it sounded a little familiar, because if the ratio is unfavorable, then
So I think the first and most important thing is that it seems that in order to have fibril formation in presence of membranes, you need to have both a population of protein, of bound protein and free protein. So when in our experiments, when we are in conditions where there's membrane excess, so we don't have much or not at all free protein, we don't see aggregation happening. So it's inhibited. So this makes perfect sense for me, that for aggregation to happen, you need to have both. You need to have high enough protein concentration to have both it, to have it free and bound. But you need to have free, at least in this simple system as we have it here. And this makes sense, I think, if you think about it. Whatever is like the nucleation event, you probably need a free protein coming and doing something, and not just proteins bound on membrane, which the proteins on membrane seem to be happy. So if they are bound on the membrane, they don't seem to want to aggregate. But when there's excess, and then things happen.
So my last question is more for my own curiosity. It's, you may know this, that the transgenic knockout works. When knocking out alpha-synuclein in mice, there's little or very subtle effects on somatoproduction.
Yes.
Which was disappointing when they first did the studies.
Yeah.
And then they have continued to combine that with knockouts of the other forms, the beta and gamma synucleins. When all are out, then you start to see it. So what about the other synucleins, beta and gamma?
Yes,
Can
so
they replace?
So one of them.
What are their properties relative to them?
Yes, so one of them, I don't know which is it, beta or gamma. One of them, I think, does lacks the NAC region, this hydrophobic region in alpha-synuclein sequence. That's what I know. And this is of course like a really big difference, right? Because this hydrophobic part in alpha-synuclein, it forms the core of the fibrils, and it's obviously very important for aggregation. So having alpha-synuclein, not alpha-synuclein, but beta or gamma, a protein which lacks this region, it's like a completely different protein. What it does, I don't know, and I don't remember what is the third synuclein, what is different in it. And it's hard for me to say whether they free, what is their task, whether it's in binding or in aggregation. And but probably I mean, it would I would definitely need to read up a little bit on how they look and how they from what's the structure, then I would could get some better idea how it binds to the membrane. But I have no way to say on which they all three could have biggest effect, on which process, or because we also don't know whether the pathology comes from but from aggregating or from some disruption of the healthy function, right? Which is
the idea here is, of course, that they could compensate physiologically.
Mm-hmm.
When this happens in other systems also, where you have several types, that the alternative type may then become more abundant and substitute.
Mm-hmm. So I know that there are similar work done with similar perspective as ours on the other synucleins. So I guess there is information that can be looked into, and we could get some idea, I guess, at least hypothesis.
From point of view of normal physiology, that could be quite relevant.
Mm.
Because, it also complicates studies on it, because if the different synucleins can substitute for each other.
Yeah. But that seems really interesting, an important point. Mm.
Okay, well, thank you very much.
Thank you. Thank you.
Okay, thank you. So finally, are there any questions from the audience? No, then if not, I suggest we thank everyone who's been participating today. And as I said before, everyone who's not on the examination board or the opponent or a supervisor should now gather in the physical chemistry coffee room for some refreshments. So see you there in a bit. Thank you.
Den första biten. Här är det. De är ju lite